Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Kyle from Business Review, and I'll be your host today. It's our pleasure to have Modality Solutions with us, who's presenting this webinar entitled Cold Chain Validation Best Practices, Including Immunotherapy. Today's guest speakers are Gary Hutchinson, President at Modality Solutions, and Daniel Littlefield, VP Engineering at Modality Solutions. I'd like to welcome you all to our webinar platform on 24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link you received via email to rejoin the session. If you want to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen, click Submit, and we'll have some time at the very end to address any questions or thoughts that you may have. Please use the yellow Help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But for now, please allow me to welcome Gary Hutchinson. Over to you, Gary.
Thank you, Kyle, for the great introduction, and thank you, everybody, for showing up and getting a chance to talk today about cold chain best validation practices. I just want to spend a little bit of time talking about Modality Solutions. We're a biopharmaceutical cold chain engineering firm, and we work with all sizes and types of innovators to design and implement and essentially validate cold chain processes. We've worked on over 75 therapies over the last 18 months, including monoclonal antibodies, antibody-drug conjugates, orphan drugs, vaccines, and some combination medical devices as well. I'm also very excited. We got onto the Inc. 5000 for the first time this year, which recognizes the fastest growing private companies here in the United States. So just to review our key learning objectives today, or why you came here today, we're going to review regulatory guidelines and how you can integrate your stability data and testing into your environmental hazards during transport and into your validation master plan for your cold chain. We're going to talk about lane qualification, how you can use your operational and performance qualification approach to qualify those lanes. I'm going to spend some time on ASTM D4169 and why the agency is starting to ask questions. Is that really sufficient testing for some of these new therapies that are out there? And lastly, we hope to give you some confidence in your cold chain and how you can put this all together for your filing and inspection and help you improve your validation strategy. I think one of the things that's really interesting in our industry right now, the progress of these new therapies are really amazing. It wasn't that long ago in 2013 when cancer immunotherapy was Science Magazine's Breakthrough of the Year. And following that, very quickly, we had our first therapy approved in Japan and here in the U.S. A year later, the FDA approved the first in a new class of immunotherapies, the oncolytic viruses. And just 2 years after that, the first antibody-drug conjugate was approved, and later in that year, the first two CAR T-cell therapies were approved as well. So I think the progress and how we're advancing on these therapies are amazing, but it's really made a challenge for us as cold chain professionals on how do we actually validate our cold chain. We included some feedback from the agencies on immunotherapy submissions just to set the table. The first thing is there's a lot of great feedback from the agencies on that we have to include all appropriate sites. You have to, and this is an exact quote, please include the shipping of all your sites that are appropriate, including your clinical sites, and make sure you look at the worst case scenarios when you're choosing those sites. The other thing that we found very interesting when it came to immunotherapy submission feedback is all that they're looking at deep frozen aggregate formation. So even though the product is deep frozen, there's been questions on do you understand and are aggregates forming during the transportation testing? Another question that comes up with a lot of our clients, and the feedback has been very specific on this, that they require drug product testing. So there's been some opportunities or some feedback that you have to do that drug product testing after the end of either your simulation to just confirm that the product is still within specification. And there's been a lot of questions on how do you select those quality attributes to assess when you're doing your drug product testing and you need the justification for that. And in all cases, you're going to have to make your case with data, that the data has to be provided to confirm everything, the justifications that you're making. I think the important thing, and it's a really great quote that I wanted to highlight here, this is directly from the agency, is while temperature maintenance is one of the critical elements for shipping, these results alone are insufficient to support shipping validation respect to product quality because other factors, vibration and or pressure changes, may significantly impact product quality. And I think this is a realization by both the regulatory agencies and within the industry that temperature alone is not the only environmental hazard that has to be considered when you're doing shipping validation, that you have to look at these other factors as well and confirm that they're not having any impact on drug product. So what it comes down to is how do you address these issues? And we're going to spend the balance of the time addressing those. But you have to make sure you can demonstrate your drug product robustness with distribution testing. You have to assure product identity, strength and purity, and quality and safety. And these environmental hazards that you choose are justified and based on some well-defined industry guidelines. You have to also make sure you assure control with your qualification strategy. And by doing that, we recommend doing equipment and packaging qualification and do that appropriately. You have to link any drug product robustness testing to your selected performance qualification transport lanes. And then you have to ensure good distribution practices are managed and you're following your PQ strategy. And then the last thing that's really important when you're putting together your validation package is you have to explain your strategy for ongoing control, especially with some of these more complex cold chains are coming out with these new therapies. The agency is very interested in understanding how you're going to assure control going forward. So that's a combination of how your quality management systems work together, your monitoring and control systems, and what external performance management you have with any subcontractors are going on. All of those need to be addressed in the submission. And the one thing we like to share with all our clients is we always recommend writing it down in your validation master plan. One of the things, and it's understood in the industry right now, but we can't stress it enough, make sure that when you put together your strategy, you're putting it down in a validation master plan that explains all these steps, because you have to make sure all of these processes are integrated together and work together to put together your control strategy. Now I'm going to turn the presentation over to Dan, and he's going to talk about some of the specifics here.
Thanks, Gary, and thank you all very much for participating this morning. It's really kind of a fun topic for me to talk about. I'm going to start out by doing a little bit of a comparison and contrast on product damage versus packaging damage. When we look at the product and package damage, the first thing we want to talk about is what the hazard is and whether the product damage that you that you get, whether it occurs during normal operation or as part of abnormal operation. And then the second part is we want to make sure that we understand the difference between product damage and primary packaging damage and the harm associated with that. So this graph is really a demonstration of damage boundary curves for a shock hazard. So the x-axis is total change in velocity, and the y-axis is peak acceleration. When we start in the kind of the lower left-hand corner, that is our operating space. And then as you move up to the right, we get into kind of that gray area, and that gray area is the vial damage boundary. So this is the primary packaging. Now, if you have damage to your vial, cracked or broken vials, it is easily visible. And that's an important factor for understanding the risk associated with it, because if you recognize that that vial is damaged or broken, you don't use it. So that's a good thing. On the same graph, if you keep going up and to the right a little more, you get into the blue area, which is our product damage boundary. And at that point, that's where shock events are actually damaging the product in the vial. Now, the good thing about the way this particular failure mode is set up is the vial is always going to be a leading indicator of product damage. So if vial is damaged, then we know that there's an issue. Product damage is much more difficult to address because it's not always visible. In fact, frequently product damage is not visible. And so by having a vial damage ahead of the product damage, we actually have a somewhat lower risk profile when we look at that. Now, I want to contrast that then to the next slide. And in this case, the positions are reversed. We're seeing product damage ahead of vial damage. And this is kind of your worst case scenario as far as product damage versus primary packaging damage. Because in this case, there are a lot of different scenarios where you could have damage to the product and not realize it. And if you don't realize the product is damaged, then whatever harm then can occur to the patient. The other thing I would note in this graph is there's a little bit of overlap between the operating space and that product damage. And so as a result, during normal operation, you will probably get some product damage. So this is definitely a risk profile that is worst case for looking at this. So finally, as the third graph, we switch back. So the vial damage is a leading indicator for product damage, which is a good thing and encouraging. And the operating space does not overlap either of the damage boundaries. So this is kind of a best case as far as looking at shock damage and the operating space and the associated damage boundary curves. So the last couple slides I was looking at was operating space for a single hazard. I'm going to take a look at a more general operating space for multiple transportation hazards. For defining your operating space, the first thing you need to do is understand your risks to your product and primary packaging, and that's where your subject matter experts come in. We'll discuss in the next couple slides about the limits and dimensions of different hazards, but you need to understand what those limits are. So, for example, if you have a protein in solution that's refrigerated, so 2 to 8 a recommended storage condition, you need to understand whether freeze-thaw is a significant issue. And if it is, you recognize that and set that limit. And the same thing on the high side is your, at what point do you start having product damage? Is it 10 degrees, 15 degrees, 25 degrees? So obviously for more robust products, you would be able to set your operating space a little differently. But you need to understand those limits to be able to define your operating space. And then finally, you need to take that information and use it to design your simulation. And you want to test the hazards that are relevant. And the converse is also true. Don't test irrelevant hazards. just to make sure we have a kind of common terminology, I went ahead and put together a slide to talk about risk. And we define a hazard as a potential source of harm, and then harm is damage to health. So we're focusing on patient, and that health damage can occur from a loss of product quality or a loss of availability. So that's also consideration as well. And then risk is a combination of the probability of the occurrence of harm and the severity. Now, for severity, we've seen a pretty wide variation. You can go anywhere from, say, loss of efficacy for a non-life-threatening health issue to loss of efficacy for a life-threatening health issue, which is obviously a significantly higher risk, or a product safety issue resulting in a serious adverse event. So then continuing along the discussion around the environmental hazards. So we look at primarily 5 environmental hazards: temperature, shock and vibration. And in this case, we included shock and vibration in the same line. They're both kinetic energy events. Shock is typically a discrete event with a higher intensity. Vibration is a repetitive event at a lower intensity. Pressure and humidity. So those are the 5 environmental hazards that we focus on when we start looking at the risk analysis and the simulation. So once you've done your risk assessment with your subject matter experts, that allows you to set the limits for each of those hazards. You're able to take that information. Now, the second piece, and that's kind of an important factor, is the environmental hazards that we talked about. They have multiple dimensions, and so there are issues around that. And once you combine all that, put it all together, what you wind up with is multiple hazards, multi-dimensional hazards, some of which have upper and lower limits. And so it's a fairly sophisticated operating space that you're looking at. And so the testing is also fairly sophisticated. And it's important to do concurrent testing as well, because some of your worst case locations in your operating space are going to be combinations of factors, of hazards and dimensions. So this table I'm going to kind of talk through. It is an example. It is not set in stone, so there are exceptions to these, but it gives you an idea of kind of how we look at the particular environmental hazards. So first, with temperature, is frequently two-tailed, meaning there's a high limit that is undesirable and a low limit that is undesirable. You might have an example where perhaps your storage temperature is less than -60 and you don't have a low temperature limit, so that's fine. But for most of the ones that we work with, we're looking at 2 limits. It's two-dimensional. It's temperature and its duration. So there's a temperature and a time component as well. So that has to be taken into consideration. For vibration, it's one-tailed in that low vibration is a good thing. So it's only high vibration we consider, but it's a 3-dimensional hazard in that we look at it in terms of frequency and intensity and duration. Shock is one-tailed in that low shock, again, is not really the issue, and we look at it in terms of acceleration and velocity change. And because it's a discrete event, we don't look at duration there. Then for pressure, one-tailed because high pressure really doesn't occur in most distribution processes unless somebody climbs on board a submarine and takes it down. But if they do, please let me know, because I would be kind of interested in that logistics process. But so pressure, typically the low end is where we have the concern, and it's also two-dimensional. You have force per unit area and a duration. And then finally humidity, which is two-tailed because very high humidity is undesirable, very low humidity is potentially undesirable, and 2 dimensions of percent of water vapor in the atmosphere, as well as duration. So when you put all that together, and I've created a fairly simple operating space here because I can't draw hypercubes. So in this case, we're looking at temperature as a hazard, and that's the x-axis, vibration as a hazard, and that's the z-axis, and then the harm associated with those, and that's my y-axis. So the vibration, as you can see, is one-tailed, so it just goes up as the vibration goes up, the harm associated with that. And the temperature is two-tailed, so it has a saddle. There's a center of the specification where we're at kind of minimum harm and then increasing in both directions. The important thing is when just looking at the graph, you can see that the highest harm locations in this operating space is your back left and your back right, which is low temperature and high vibration, and high temperature and high vibration. Now, when I overlay testing strategies onto this operating space, it kind of creates an interesting picture. Let's start out with kind of the basic things that we look at. So your end of shelf life studies are going to be where the green dot is. It's going to be at the optimum temperature and no vibration. Important thing needs to be done. So that's one place that you're going to investigate. And then there's a high likelihood that you're going to do a freeze-thaw study, which will end up on your on the low temperature side and low vibration. So that's that yellow dot in the lower left-hand corner. And then you're also probably going to do accelerated stability studies, and that's going to be the low vibration, high temperature side. So you've kind of covered that edge pretty well. Now, I want to take a moment to talk a little bit about ASTM D4169. So first thing you can do with D4169 is you can test at ambient conditions, and that's the blue dot on the high temperature side. You can also do D4169 in the thermal packaging, so in controlled temperature, and that will give you the blue point that's kind of in the center of the temperature and up a little. The problem with ASTM D4169 is they have accelerated vibration profiles, and the accelerated profiles, they're great for package testing, but they're not optimum for product testing. And so you have an accelerated profile that is not consistent with what the product is going to see in the real world, so it's not realistic. And so for something like distribution cycle 13, they recommend 120 minutes of airplane and 60 minutes of truck. And the thing I can promise you is there are no products in the U.S. that get from manufacturing to patient with 2 hours of airplane and an hour of truck as their worst case. And that's why they're positioned so low on the vibration, because they're well away from the worst case locations. And that's why we recommend, because of the way the operating space plays out, to set up a concurrent multivariate simulation. And it looks at low temperature and high vibration, and it's unaccelerated real-time simulation, and that gets you the orange dots in your back left corner and the back right, which are your worst cases. So that's how we end up with the transport simulation that we recommend. Now, once you've completed the simulation, you basically come out with three important points. Number one, you've looked at your transport lanes and you've ensured that your operating space fully encompasses all of those. So that's the first thing. So you know that you're looking at your worst case for your transport lanes in this in this simulation operational qualification. The other thing you've done is you've concurrently tested. So you've looked at the worst case limits. You've looked at the longest vibration and high temperature, longest vibration, low temperature in your operating space. So you've looked at those worst case limits, and that really has, once you've demonstrated good product quality post simulation, you've demonstrated that your product is robust for your worst case exposures, and that's the important point that the regulatory authorities are interested in, is to be able to make that statement. kind of a key part of your validation master plan. So because of the way we recommend the distribution operation qualification, it allows a little simpler strategy for your performance qualification. And so the key things that we want to demonstrate in the PQ is, first, we want to demonstrate that we're operating in a state of control. So we do that by, when we execute the PQ, by getting a robust outcome that is in our normal operating range. So that confirms, number one, that the lanes that you're conducting the PQ on are within the operating space of your OQ, and it also demonstrates that you're operating in a good state of control, that you're not having issues with procedures or equipment. The one thing the PQ does not do is it does not investigate the worst case. You could call your carrier if you want to and say, hey, when you pick the box up, can you be sure to drop it, a few times, 36 inches, and drive around on a lot of bumpy roads, for an extra few hours to kind of get that worst case. And some of them might do it just for fun. But the fact is, even if you do those things and you still demonstrate good temperature, then you still haven't looked at your worst case. So there's really no realistic way to make a PQ anything other than somewhere in the middle of your operating space, which is exactly what we recommend for our PQs, because the OQ has demonstrated the worst case performance, and that's really where you the right way to focus it. So now that we've talked about the operating space and the qualification strategies around the product, I want to talk about some of the specific factors and concerns for different treatment types. First, monoclonal antibodies. So the focus there is on the robustness in concurrent realistic simulation. So that's really the interest. The interest, as Gary alluded to in one of the early slides, it's temperature plus other stuff. So you need to understand what the other stuff is and include that in your considerations. The most common hazards we test for monoclonal antibodies are shock, vibration, and temperature. Now, if it's a prefilled syringe, then pressure is normally added as well. So another important consideration there. If you're working with a lyophilized product, there are some additional challenges because the cake integrity and its relationship to potential harm is a hotly debated topic. So that's something that your subject matter experts have to be able to address as you go through that for the lyophilized products. We recommend PQ as a post-approval commitment because we really want to do the PQ on the commercial process, and you really need an approved product to have your fully completed commercial process. The important thing, though, is you do want your protocols ready ahead of time, because the FDA is likely to ask for them in a PAI or as part of an information request. So you want your protocols ready, but we normally recommend PQs as a post-approval commitment. The PQ should include all your process steps. So manufacturing to fill finish to label pack to distribution are all steps where a PQ should be done. But you don't have to do all the lanes. So especially when you're talking about, say, distribution to regional distribution or specialty pharma, those could be 30 or 40 or 50 lanes. And the important thing is to demonstrate that the lanes that you test are within your operating space and operate in the state of control, but you don't have to do all of them. And there's no expectation for drug product testing in the PQ because you've already demonstrated good product robustness in a worst case scenario in the OQ. Antibody-drug conjugates so are handled a little bit like monoclonal antibodies on steroids. So the precursors should be included in validation. So it's typically the monoclonal antibody itself plus the linker plus the toxin. So for the precursors, the monoclonal antibody, you would address it just like we talked about in the previous slide. For the linker and toxin, they're typically small molecule. They're typically pretty robust, and so they're usually not tested in simulation. You still need to obviously include them as part of the validation master plan and include them as part of assurance of temperature control and those sorts of efforts. And then finally, the antibody-drug conjugate itself, which is the product of the MAB plus everything else, also needs to be addressed much as you would a monoclonal antibody. And then finally, I want to talk a little bit about cell therapies. So your validation master plan needs to address the same three big issues that you would for other products. Number one, you want to demonstrate that your product is robust in distribution. Number two, you want to demonstrate assurance of control with the qualification strategy you've adopted, and you want to demonstrate that you have a strategy for continuing control. Now, cell therapies have typically a much more elevated risk profile, especially autologous cell therapies. And that ties back to the nature of the severity of the illness. Because it's autologous, the precursor, the apheresis, is limited in supply, and if it's lost or damaged, or if the product is lost or damaged, there's no shelf to go back to get the treatment. So that increases the potential severity of the risks because of that. Also, for many of the cell therapies, there are pretty tight treatment windows as well. And so all of those things combined increase the risk profile when you go through and do your risk assessment and your consideration in validation. So that's a big factor that we've seen with the cell therapies. Now, the cell therapies we've worked with are typically stored in liquid nitrogen temperature, so roughly minus 190, and they are one-sided specifications, and typically no flexibility on those limits. So we don't normally include temperature variation as part of the simulation for cell therapies. There are also some technology issues with testing things at, say, minus 100 centigrade, for something that you would say has a specification of minus 160, less than minus 160. Now, the precursor for cell therapies is the apheresis, which is collected, and that needs to be included in your validation master plan as well. So we recommend that. Now, the apheresis, because it's cells with cell wall intact, are typically more stable than most of the other things that we've talked about here today. So they are normally not included as part of a simulation strategy, but the assurance of temperature control is still obviously important. And it also ties back to the fact that because it's a very limited precursor, there are elevated risk profile issues if you were to have a temperature excursion. And then finally, to talk a little bit about the final drug product. So once you've done simulation, you want to test a product to confirm that it's still robust. And we typically see cell viability and purity, impurity testing as typical for the autologous therapies. So that wraps it up for me. Thank you very much for everyone's time. I'm going to hand it back over for Gary to wrap things up.
Thank you, Dan. I think you covered a lot of material there really quickly, and we're absolutely going to be able to be available to answer all the questions that are coming in right now. What we wanted you to understand through this through the webinar is these new therapies require new approaches to your cold chain validation strategy. Regulatory requirements are changing. I think one of the things that Dan talked about, especially for the ADC class of therapies, there's a lot of testing going on not only the monoclonal antibody and then the final drug product, but also on the precursors as well. And I think it's an important takeaway on that, plus some of the challenges that comes with product testing for the gene therapies, specifically the T cells. It's really important to understand your damage boundaries. Dan walked us through the damage boundaries for a single hazard, the shock events that are there, but you have to understand what those damage boundaries are and define them very clearly in your operating space for all the hazards that could potentially impact your therapy. And once you understand those damage boundaries and what those hazards are, you have to make sure you're testing within your operating space. And I think the agency feedback has been clear, and as Dan walked through, testing in your operating space has to be something you do concurrently. There's hazards, there's therapies out there that are very temperature sensitive, but are also sensitive to other hazards out there. And the only way you can really test your operating space to the expectations of the regulatory bodies out there is test your operating space with these concurrent hazards. And the last thing, I think we walked through some of the main classes of the immunotherapy products that are out there. You really have to make sure you vary your approach by therapy. One size does not fit all. And depending on your therapy and depending on your drug and its specific risks to these environmental hazards, you have to take care up front to understand what those hazards are and make sure your validation approach matches your therapy and your cold chain. As I mentioned, Dan and I are going to be available now for questions that are coming in. We're going to answer everyone we can with the time that we do have. But if you need to get a hold of us, we're always ready to have a conversation with you about your specific issues. You can reach us by email, info@modalitysolutions.com, on the phone, or head to our website and you can get a hold of us through the contact page, and we'll reach right out to you. We're going to go on speaker now to start answering all the questions. Kyle, we'll turn it back over to you.
Okay, thank you very much. It's time for the Q&A. So a quick reminder on how this works. Just type your questions into the box in the top left-hand corner of your screen, click submit, and then wait to hear if your question is read aloud. I've already had a few come in here, so we can just get started. Our first question today is, on your discussion on ADC, you recommend testing both the MAB and the final drug product. Is this necessary? Isn't that redundant testing?
So, great question. So the concern from the regulatory authorities and obviously from a technology perspective is you want to make sure that the monoclonal antibody is good, is not damaged before you create the ADC. So you need to have a test first for that. And because the monoclonal antibodies are frequently go through a pretty severe transportation process before they go to the ADC for completion, it's critical to make sure that the product is still good to do that. The second part then is once the product is completed and you have the DP, it's the same issue, right? Because the DP then goes out into another distribution process, which is different and needs to be assured as well. And that one is, I think, especially true because typically once these products are shipped, there's no additional quality testing done. And so it's really important to make sure that as part of your distribution simulation, you ensure that the product is robust coming from manufacturing.
Yeah, and I think Dan hit on all the points. I think the thing, I think we were a little surprised that was the request, that the monoclonal antibody as a precursor and the final drug product were both going to be tested and tested very similarly. So, part of that was agency feedback, but I think there's some really good scientific justification for doing that testing that way. So, part of it is just good engineering and good science, and part of it was regulatory expectations on some of the projects we did work on.
Okay, thank you. Another question here saying, I understand the damage boundaries as a concept. However, the team is really worried about glass breakage. How do you ensure no vials will be broken during testing?
So, another good question, and the fact is, because we test pretty robustly during our simulations, we on occasion have had broken vials. We don't view that as a failure because, again, we're pretty aggressive on where we test versus the operating space. So we understand and accept that perhaps some vial breakage will occur during the simulation. If there's a significant issue with vial breakage in simulation, then that typically means that some additional work might need to be done on secondary packaging to ensure protection of the primary packaging, and so that's the logical next step there. But we never really seen that. So, and most of the time we don't have vial breakage, and so it doesn't really become a concern.
Okay, very good. Question here asking, can you comment on bulk drug transport validation for frozen drug substances? It's a whole lot different than DP.
Yes, and it's an excellent question because we frequently see bulk drug substance transported frozen, and frozen is a bit more stable than protein in solution, and then it ends up being formulated into protein in solution as DP in vials. So what we actually do is we set up our simulation to choose the worst case, which is DP in a parcel environment, and we include the bulk drug substance moves in the simulation, so that we encompass the entire supply chain, including the bulk drug substance, but we test it as a worst case. So that's usually the way we carry that study out.
Yeah, and it's a really good question because I think there's a little bit of a change here in the agency's thinking where frozen drug substance was seen as really not an issue as long as the product remained frozen. There seemed to be no challenges as far as particle formulation or any of those type of issues, and there was some downstream processing that was going to go on. But in recent filings, there has been agency questions about how do you know there isn't going to be particle formulation even with frozen drug substance shipping. So our recommendation is you have to come up with a test plan, and it's going to require testing that frozen drug substance in some type of simulated environment and then confirming afterwards that particle hasn't formed even though the product is frozen. We're, from a science or from an engineering point of view, we're not completely on board with that. Frozen drug substance has a very limited risk, we believe, for a particular formation, but this was actually in a request for information from the agency specifically about drug product frozen drug product and particulate formation. So it's something you do definitely have to consider.
Okay, very good. Someone here is asked if you can tell us when this activity needs to be completed at what stage of development, as in before phase 3 or during 3 validation lot production or just prior to BLA submission.
So that's a great question, and so our recommendation is you should start putting this work together probably very late in phase 2 or during phase 3. The entire process of doing this takes 3 to 6 months, depending upon availability of product for simulation, test schedules, and things like that. So to make sure you're off the critical path for submission, it's best to try and be working on it in phase 3. Now, that's not to say that we haven't had people occasionally come in and say, "Oh yeah, we're submitting our BLA in 6 weeks, and we need to do this," which requires kind of an all hands on deck sort of strategy. So, but I think you start thinking about it in early phase 3. That puts you in pretty good shape, and it also allows you, if you have questions or concerns, you can put together questions for the regulatory agency to confirm that your validation strategy is acceptable.
Yeah, timing is always a question. Our recommendation is if you're going to do any type of testing before phase 3, that testing is really on formulation development. So when you're doing the formulation design, you're coming up with your final commercial formulation, doing some sort of screening test on different formulations you're considering always makes sense. But you don't always have time to do that. I think the compressed timelines that Dan was talking about are primarily what we see with fast track or orphan drug consideration, where the timelines are so accelerated, which is a positive thing, that the agencies are allowing, showing a fast track and compressing that timeline for patient populations who really don't have other alternatives that are out there. But it makes this testing schedule a lot tighter when we get involved in that type of testing. And many times it's you're making commitments to do the testing, and it ends up sometimes being almost a confirmatory testing at that point because the timelines are so reduced. I think the other issue really comes in on the testing is driven by drug product availability as well, and finishing up the specification and any analytical methods that you're going to do for release testing. That all impacts timeline as well. So I think overall we're saying start as early as possible. You're going to have to start with your commercial formulation for the validation package, but a lot of it is driven by not only filing timelines, but also from drug product availability as well.
Okay, thanks. Question here saying ASTM 4169 has been an industry accepted standard for years. Are you saying it's no longer valid?
So ASTM is actually D4169. So ASTM D4169 is a package testing standard and has been around a long time. So it's an industry recognized standard, and so it was quickly adopted by the biotech and pharma industry because it was out there to kind of get an idea of how the product is affected in distribution. The problem with it is it's not a great simulation, and I think we talked about that a bit in one of the slides. So the regulatory authorities have started to recognize the problems with it, and so they're pushing back. I'd say we've seen at least 7 or 8 rejections of D4169, basically saying it's not an acceptable methodology for simulation in the last 2 years. Is that about right?
Yeah, 2 years probably.
So.
And I think just to emphasize Dan's point, that 4169 was designed to and is best used for package testing, and it came out of a package testing environment. And it may have been perfectly adequate for some of the early biologics that came onto the market, but for these complex therapies that we were emphasizing today, the testing methodology doesn't really meet the requirements for the challenges that the drug product is going to see in transit and what you're going to need to do to prove drug product quality, efficacy, purity, all the way through the cold chain to the patient.
Okay, thank you very much. I think that about does it for today, which just leaves a big thanks to Gary and Dania for what a great presentation, and of course to Modality Solutions for sponsoring this session. To everyone watching, you're going to receive an email shortly letting you know how to access the on-demand version of this webinar. Or you can access this through our website, which is www.business-review-webinars.com. Thank you all once again, and I hope everyone enjoys the rest of their day.